This presentation is about GCSE microscopes. Here are the key questions. What determines the level of detail that we can see with a microscope? What has the development of the electron microscope allowed us to do? What units are used for very small sizes? Today we'll be working on the skill of unit conversions. Before learning about the microscope, it's important to know something about the history. In 1665, a scientist named Robert Hooke discovered cells. He used a microscope which had two lenses, and it magnified the image by about 30 times. Later on in 1675, Antony van Leeuwenhoek used another microscope to discover animalcules. His microscope was better because he invented lenses which showed things at a much higher magnification. Finally, in the 1930s, and still used today, the electron microscope was invented. The electron microscope changed microscopy because it allowed us to view cells in much greater detail. It would magnify the image so much that we could see inside of each cell and all of the organelles there. Robert Hooke was one of the first people to examine cells using a microscope. He examined bark from a cork oak tree and saw little box shapes. He called them cells because he thought that the boxes looked like the small rooms that found in monasteries at the time. Hooke realised the importance of making accurate drawings of what he saw to help explain his work to others, and the picture here shows drawings of cork cells which were published in 1665 in his book Micrographia. Antony van Leeuwenhoek discovered animalcules. Animalcules were first observed in rainwater. He saw tiny organisms and named them animalcules, and because he was so fascinated by his discovery, he searched for other ones in different places. Here are some drawings of animalcules that he found in scrapings from his teeth. We would call these bacteria. Instead of light microscopes, we now use electron microscopes. Electron microscopes will use beams of electrons to pass through a specimen which will build up an image. These microscopes can magnify up to 2 million times. Van Leeuwenhoek's, on the contrary, could only magnify to about 270 times, and Robert Hooke's microscope could only magnify to about 30 times. These are the parts of the light microscope. The eyepiece lens is where you look through and magnifies the image once. The objective lens magnifies the image again. When you combine the two magnifications, this is called a total magnification, which we'll talk about later. This is the stage where you put your slide, and these are the stage clips, which hold the slide steady. The light source shines a light upwards through the specimen and through the two different lenses so that you can see it. The coarse focus will move the stage up and down, and this will bring your image into focus, and the fine focus is used to make very small movements in order to clarify your focus. So let's work out how to calculate a magnification of an image. We use the equation total magnification equals the eyepiece lens multiplied by the objective lens. For example, a microscope with an eyepiece lens of times 10 and an objective lens with a times 20 would magnify the image times 200. 200 is a product of 10 and 20. On the objective lens, the magnification will be printed. Here's an example of calculating total magnification. A microscope has an eyepiece lens of times 5. It also has 3 different objective lenses, times 5, times 15 and times 20. Calculate the 3 total magnifications for this microscope. Pause the video while you work this out. Here are the answers. 5 times 5 will give you 25, 5 times 15 75, and 5 times 20 will give you 100. So the 3 possible magnifications for this microscope are times 25, times 75, and times 100. Sometimes you'll be asked to calculate an unknown. For example, this is a picture of a ruler. It's 50 centimeters long in real life. If the ruler was 1 meter long on the whiteboard, then what's the magnification number? Well, we know that it's 100 centimeters, which is a meter, being my image size projected onto the whiteboard. The real size is 50 centimeters. It's only half a meter long. So therefore, the image on the board must have been magnified 2 times. Let's talk about the concept of resolution. We can see that the 2 images are the same size, but the one on the right is at a higher resolution. Resolution means the smallest distance that 2 points can still be seen as 2 different points. Higher resolution means a more detailed image. As microscopy has developed, resolution has improved. We now have better lenses, we can stain our specimens better, and also there is development in light sources. All of these contribute to more detailed, clearer, and higher resolution images. SI units are the international system of units used in science. We refer to them as SI for short. The units for distance are based on meters. You may recognise these from your mathematics lessons. We use millimeters, micrometers, nanometers, and picometers to describe units each 1,000 times smaller than the previous. Of course, you can use standard form to show this rather than using decimals, as these are very long numbers. But you have to be able to do a conversion from decimal to the standard form and vice versa. If you're unsure how to do this, please ask your teacher. Here's a conversion example. The diameter of the red blood cell is 0.000012 meters. Express this in micrometers. We understand that 1 micrometer is 1 times 10 to the minus 6 meters. So if right at the number, we have to jump the point 6 times backwards. This gives us a value of 12 micrometers. So for the next activity, try to copy out this table and convert the lengths on the left-hand side to standard form in meters. I've also included 2 standard forms at the bottom for you to convert back into length for extra practice. The answers will be on the next slide, so please pause the video and have a go at these. Okay, so hopefully you got these right. If you didn't get these right, it would be a good idea to get your teacher to sit through with you how to do the conversions. For more information or for more calculations, please check out the other video on microscope calculations.